Hey DBU, welcome to our online lecture again. This week we're gonna continue where we left off with cancer and weave that into mitosis and meiosis, which is cell division. So again, our schedule is a little bit different, a little bit rearranged. So please go onto our Blackboard course schedule and see where things have changed. We'll try and cover all of this in the online lecture. We'll go over it a bit on Tuesday and also talk about reproduction and human development. All right, so let's revisit cancer real quick. So we saw that cancer, all that cancer is, are cells that divide in an uncontrolled manner and result in a tumor. So that word divide is bolded, and that's what we're going to be looking at in this lecture is cell division. And in this slide, we looked at how cancers arise. So we start here with normal cell division, which is happening all the time in most of the cells of your body. And eventually, during cell division, one of the cells is going to accumulate a mutation that will either disable the brakes of the cell cycle, which are tumor suppressors, right? They prevent or inhibit the cell cycle from happening, cell division from happening if there are errors, or a mutation that will activate the brakes, a mutation that will activate a proto-oncogene into an oncogene, which will encourage cell division. And so this cell will continue to divide in an unregulated manner and become a tumor. So again, cancer really, at least in simple terms, boils down to uncontrolled cell division. And then next we actually saw how cancers have to disrupt the cell cycle in order to become a tumor. And so this is where we're going to pick up with our lecture today. In the cell cycle we have mitosis. And mitosis, again, is cell division by all means. And you might ask yourself, why should I care about mitosis? Why are we looking at this process of cell division? Well, for one, this is what explains how you went from one cell when you were an egg that were fertilized by a sperm to now 37 trillion cells, which make up your whole body. This was 37,000 billion times that your cells have divided. Unbelievable, right? So mitosis explains how you went from one cell to 37 trillion. And it also is important for wound healing. So whenever you cut yourself, the ability to scar and to regrow a piece of skin that you've injured, or even a piece of liver that you might have injured, is thanks to mitosis. And then obviously cancer, right, is the pathological tie-in. So cancer is a disease, a proliferative disease, a disease where you have proliferation or really upregulated cell division. All right, so let's look at mitosis more closely. Let's revisit the cell cycle first. So you have interphase, where the cell grows and the genome is copied or duplicated. You have mitosis, where the genome copies separate. So in mitosis, even though it's similar, it's analogous or interchangeable with cell division, it's not actually cell division. This is really more where the copies of DNA separate into two nuclei. And then there's an actual process where the cells pinch off called cytokinesis. And kinesis means movement, cyto means cell. So it's like cell movement. And this is where that cells actually divide. And then you start the cycle all over. So let's look at interphase. Interphase, you have three parts. The first one is G1, which is where growth is happening. And then you have the S phase, where the DNA is being duplicated. You have the G2, the gap 2 phase, where the cell is preparing for mitosis. So it's doubling other things, getting ready for that division process. And then finally, now you have mitosis and cytokinesis, where the cell divides. So what cells undergo mitosis? Somatic cells, which are basically every cell in your body except for sex cells. And these cells are diploid, which means they have two sets of chromosomes, which is why you have di in there. Di always means two. So in the human body, you have 46 chromosomes. Basically, in every cell, you have 46 chromosomes. And it's really 23 pairs of chromosomes. Again, remember, a diploid cell has two sets. So you have two sets of 23 chromosomes, which comes to 46. You have one from your dad and one from your mom. So in the picture to the right, imagine that the red chromosome is from your dad and the blue chromosome is from your mom. So you can see in the picture on the left of that, you have chromosomes numbered from 1 to 23, and chromosome 1 has 2. There are 2 chromosome 1s. It's a pair. One of them is from your mom and one of them is from your dad. And so in mitosis, what happens is one of these cells that has 46 chromosomes will divide into 2 cells with 46 chromosomes. So you're basically just doubling the cell, right? So the way you do this is you go through interphase, where the DNA is copied. And so now you go from actually 46 chromosomes to 92. You double it. You go through mitosis, where the DNA is split into 2 daughter cells. And then cytokinesis, where the parent cell is pinched off. The cells are pinched off into 2 cells. And so now you end up with 2 cells that have 46 chromosomes each. And what about sex cells? So we said that all cells, all somatic cells undergo mitosis. So sex cells, they don't undergo mitosis, they undergo meiosis, which is when one cell with 46 chromosomes divides into 4 cells with 23 chromosomes. And we'll look at that once we get into meiosis. So let's look at mitosis up close. There's 4 steps to mitosis. So now if you'll notice in the cell cycle, we're looking at just the mitosis portion of the whole process. And there's 4 steps. You should try and memorize these for sure. So you have prophase, then you have metaphase, anaphase, and telophase. And you can come up with your own little acronyms for how to memorize this, right? PMAT or whatever, so that you can memorize prophase, metaphase, anaphase, telophase. If any of you guys speak Spanish, you could do a cool little sentence that I learned when I was in high school in Peru. It's promete Ana telefonearme. And so it brings us together and makes an actual phrase. So let's look at prophase. Actually, before we enter into prophase, right now what's on the screen is the end of interphase, where the DNA has already been replicated, right? And centrioles have also been doubled, and centrioles, we'll look at what those are in a second. But basically the DNA has been duplicated and the cell is ready to undergo cell division. So you enter prophase. And prophase, the main things that happen in prophase, there's 2 things. One, DNA condenses into chromosomes. So it goes from this big ball of spaghetti in the previous step and then condenses into these X's, which are really the pair of each chromosome that are kind of tethered together at the center. And then the second thing that happens is that the nuclear membrane, the nuclear envelope, begins to break down. And also the centrioles are going to each end of the cell and releasing these fibers, these microtubule fibers. So the next step in metaphase, the nuclear envelope now is gone and the chromosomes align in the center of the cell. So really what happens in metaphase is chromosomes align. And you can see there that the centrioles are now at opposite ends and they're releasing these spindle fibers which are attaching to the center of each chromosome pair. And then anaphase, these spindle fibers that attach to the center of each chromosome are going to pull away toward each end of the cell and pull that chromosome pair apart. So it's going to take, for example, in that first chromosome pair, that one chromosome from your mom and that one chromosome from your dad, and pull those to the opposite ends of the cell. So if you want a keyword for anaphase, it would be separate. And then in telophase, you basically redo what you did in prophase, where you reform the nuclear envelope. So now you can see that you have two nuclei, and also you start decondensing the chromosomes into spaghetti, right? And then in cytokinesis, you actually pinch the cells off. You pinch the dividing cell into two individual cells. And then you're ready to go into interphase again, where the cell is growing and doing its business. Okay, and remember, we looked at this in our last lecture, that mitosis has checkpoints. It has these security checkpoints, right? Like the TSA airport security. So in mitosis, you have two checkpoints, which ensure that things are going according to plan. And if they're not, then mitosis will stop and the cell will try to repair the errors. And if it can't, then the cell will commit suicide. So it won't actually divide incorrectly and pass on mutations to other cells. And I recommend you watch this video. It's really helpful. You'll see these steps of mitosis happen in motion. So look this up, watch it, be comfortable with it. We'll go over this in class, but I want you to definitely see this happen in motion. Okay, so now let's go into meiosis. And meiosis is a special type of cell division that creates haploid gametes. So let's try and understand what that means. A gamete is a sex cell, and you can have either sperm or eggs, right? And sperm and eggs are always haploid. What that means is they have half the amount of chromosomes that a normal cell has. Remember, a normal cell is diploid, 2 sets of chromosomes. Sperm and egg are haploid, so they only have 1 set. The HA in haploid you can think of as half the normal amount. So what meiosis does is it creates the haploid sperm and the haploid egg. And then the haploid sperm here with 23 chromosomes and the haploid egg that also has 23 chromosomes can then fuse together via fertilization to create a diploid zygote. So this is why meiosis is important, because it has to create haploid cells, right? A haploid sperm and haploid egg in order for those to fuse and form a diploid zygote, which is just the name of that fertilized egg. So it's the two genetic contents of the sperm and the egg fusing together to form a new cell. And so that is why meiosis has to create haploid cells, and why it does it differently, in a different way than mitosis. So the meiotic cell cycle is similar to the mitotic cell cycle. It has interphase, where the cell grows and the genome is copied, but then it has 2 rounds of division. It has meiosis 1 and meiosis 2, and then it goes through cytokinesis and splits the cells into 4 cells. So meiosis has 2 rounds of cell division. That's one important way it's different than mitosis. And again, meiosis, just remember, you're ending up with a haploid cell which has only 1 set of chromosomes, and these are the sex cells, either sperm or egg. And in meiosis, you're dividing 1 cell with 46 chromosomes into 4 cells with 23 chromosomes. So out of 1 normal cell, you're getting 4 haploid cells, right? Which is why there are 4 arrows that are at the end of the meiotic cell cycle. And so side by side, here's mitosis and meiosis together. Mitosis makes 2 diploid cells, each with 46 chromosomes. That's why you have 2 arrows. Meiosis makes 4 haploid cells, each with 23 chromosomes. So this is where the main difference lies between meiosis and mitosis. And the reason that you end up with 4 haploid cells is because you do an extra round of cell division. All right, so let's take a closer look at meiosis. It starts at the same place that we started with mitosis. You start with at the end of interphase. Now, I told you guys that meiosis goes through 2 rounds of cell division. So let's go through the first one real quick. You have prophase 1, metaphase 1, anaphase 1, and telophase 1. So this is actually exactly the same as mitosis. Well, not exactly the same, but it goes through the same steps. And so now let's start with prophase. In prophase, same thing happens as in mitosis. The nuclear envelope starts to break down and DNA condenses into chromosomes. So you can see there the little hand is pointing at something unique that only happens in meiosis called crossing over. And we'll look into what that is in a little bit. Then in metaphase 1, the chromosomes align in the center, just like they do in mitosis. And in meiosis, though, you can have random alignments of chromosomes. And again, we'll go into what those mean. Then you have anaphase, where those chromosomes that are in the center are separated. And then telophase, and cytokinesis, where the cells now, the nuclear membrane reforms. In this case, the DNA does not decondense, it stays condensed, and then in cytokinesis, the cells pinch off. So now we have now we're going to start a second round of cell division. So we're going to have the same 4 steps: prophase 2, metaphase 2, anaphase 2, and telophase 2. So after cytokinesis, the first round of cell division, we start again. And so prophase 2, you can notice that the DNA is already condensed. So actually, you're still going to break that nuclear envelope down, and the centrioles are going to start aligning to the ends of the cell. In metaphase 2, the chromosomes align at the center, and the centrioles send out their spindle fibers, those microtubules, that attach at the center of each chromosome pair. In anaphase, those fibers begin to pull the chromosome pairs apart, so the chromosome pairs separate. And then in telophase and cytokinesis, the nuclear membrane or envelope reforms, the DNA begins to decondense, and the cells pinch off. And again, I'm putting a little video link here that I really recommend you watch that animates this whole process for you. This is a 3D animation. It's really cool, and it'll be very helpful for you to understand how these different steps happen. And again, just while you're watching this video, notice that there are 2 rounds of cell division, one of the major differences between mitosis and meiosis. So meiosis has 2 major goals. The first one is generating haploid gametes, which we already looked at. But it needs to create sperm and eggs that have half the amount of normal genetic material, so that when they come together, they can make a normal cell, a diploid cell, that will eventually become a human being. And the second major goal is generating genetic diversity. Genetic diversity is simply variation in offspring. Genetic diversity explains why we don't look like our siblings. So if you have a family of 5 siblings, every single one is going to look different. Actually, your parents could have billions of kids and not one would look the same. And this is because of what happens in meiosis, and we'll learn about that here. But this is important, genetic diversity is important because variation in offspring increases our chances of survival under stress. This is one of the advantages of sexual reproduction, actually, because bacteria, they can only make genetically identical offspring, right? All of their offspring are going to have the same exact genetic makeup as the parent. So if some environmental stress comes along that is not favorable for that genetic makeup, all of the organisms are going to be wiped out. Whereas organisms that reproduce sexually and undergo meiosis, if environmental stress comes, then it might affect a certain genetic makeup and wipe those out. But because all of the offspring are different, some of them will survive. So meiosis generates genetic diversity in two ways: crossing over and independent assortment. So let's look at crossing over first. It's important to note that each chromosome you have exists as a pair. We've already looked at this, but we'll recap it again. So you have one chromosome from your mom, imagine that's the red one in the image, and one chromosome from your dad, which is the blue one. And each chromosome, so say this is the pair for chromosome 1, the homologous pair, each chromosome in that pair is going to carry the same set of genes for the same traits. So imagine A is the hair color gene, B is the eye color gene, and C is the skin color gene, for example. So they carry the same genes, but they can carry different alleles. And an allele is a different version of the same gene. So for example, that can be like the blue eye color gene and the brown eye color gene. Same gene, just different version. Now remember, during S phase, the DNA has been duplicated, right? And S phase will happen in mitosis and in meiosis. So in meiosis, the DNA is duplicated, so that means you have replicated homologous chromosomes, which are these little X's that you see in these pictures of meiosis. And what these are is now the red chromosome from your mom is actually duplicated and joined at the center, and the same thing with your dad's chromosome in blue. So let's look at this a little closer. These are the replicated homologous chromosomes. They're connected at the center by centromeres. So on the left, in red, you have the replicated chromosome from your mom, and on the right, you have the replicated chromosome from your dad, right? For example, say this is chromosome 1. Now notice that there's sister chromatids, and sister chromatids are the replicated chromosomes from either your mom's chromosome or your dad's chromosome. So, for example, in red, you have two sister chromatids that are attached at the center. And notice that they have exactly the same alleles. So capital A, capital B, lowercase d, right? They're exactly the same between the sister chromatids. But the homologous chromosomes may carry different alleles. So in your mom's replicated chromosome, you have capital B, but in your dad's replicated chromosome, you have lowercase b. But notice that in your dad's chromosome, in blue, the sister chromatids share exactly the same alleles. Capital A, lowercase b, lowercase d, right? So during prophase 1 of meiosis, replicated homologous chromosomes condense and pair up. So this is an image from when we looked at meiosis in detail. This is prophase 1, and you can see that the X-shaped replicated chromosomes are crossing over, have paired up and are kind of doing this weird dance. And what that is as if you took two chromosomes, twisted them together, pulled them apart, and now you end up with two chromosomes that are a combination of the original two. So crossing over can create a new combination of alleles on two of the sister chromatids. So let's see how we think this actually happens. So on the left, you have a replicated homologous pair, right? One from your dad, say we'll keep the colors the same, so the blue one is from your dad, the red one is from your mom. And if you look at your dad's chromosome, you can see that it's replicated. So there's two sister chromatids that are attached at the center by the centromere. So this homologous pair, the blue and the red, are going to pair up. And in the next step, crossing over will occur between the genes B and C. And you'll end up with a sister chromatid on your dad's replicated chromosome that has a little bit of red from your mom, and the rest of it is the blue from your dad, and a sister chromatid on your mom's replicated chromosome that has a little bit of blue, and the rest of it is red from your mom. So basically, now you've created a new combination of alleles, right? If you look at the sister chromatid on your dad's replicated chromosome, instead of having uppercase A, uppercase B, uppercase C, now you have lowercase a, lowercase b, and uppercase C. So this is now a new combination of alleles. And the same thing on the sister chromatid on your mom's replicated chromosome. Instead of having all lowercase alleles, now you have actually uppercase B, uppercase A, uppercase B, and the rest of them are lowercase alleles. So this too is a new combination of alleles. And these are called recombined chromosomes. So at the end of meiosis, you actually go through another round of cell division, right? Meiosis 2, and then you end up with haploid cells, and some of them will contain these new recombined chromosomes. So crossing over is like shuffling the alleles between homologous chromosomes, between your mom, the chromosome you got from your mom, and the chromosome you got from your dad. And this shuffling leads to genetic diversity, which is a good thing. Genetic diversity or genetic variety is good. This is, again, what creates variety in offspring, variation in offspring, which will help the organism or the species survive under conditions of stress. So let's look at what would happen if we didn't have crossing over occur in meiosis. So in this first figure, you see that little hand pointing at the crossing over event in prophase 1. And now basically you have two shuffled or recombined sister chromatids. And by the end of meiosis, you're going to produce your 4 haploid cells. And in this case, you're going to have 4 different haploid cells. No two are alike, right? And this is because you have 2 haploid cells that have shuffled or recombined chromosomes. But if crossing over did not occur, you would end up with only 2 types of gametes. So now you haven't shuffled 2 of the sister chromatids, so you end up with 2 gametes that have the exact same sister chromatid from your mom, and 2 gametes that have the exact same sister chromatids from your dad. So they are basically, for all intensive purposes, identical, the 2 from your mom and the 2 from your dad. So if you don't have crossing over, you're not going to have recombination, and that's going to lead to less genetic diversity, right? So instead of having 4 types of gametes, now you have 2 types of gametes. And that's not a good thing. That will, again, lead to a decreased ability to handle stress and changes in the environment. Okay, so let's look at the second way that meiosis can generate genetic diversity, and that is independent assortment. So now this is happening during metaphase 1, where homologous pairs are aligning at the center. So you can see there that the homologous pair, the red and the blue, have aligned at the center, and they're going to be ready for anaphase, where they are separated. So the chromosomes are separated. Now we're actually looking at 2 homologous pairs, say homologous pair 1 and homologous pair 2. And so these are separated, and you're going to end up with 4 gametes, as you always do. One cell will divide into 4 haploid cells. And so these 4 haploid cells have 1 sister chromatid from chromosome 1 and 1 sister chromatid from chromosome 2. And essentially you have here 2 combinations of chromosomes. On the top you have chromosome 1 and 2 from your mom, the red ones. And on the bottom you have chromosome 1 and 2 from your father, the blue ones. So in this example, you really only have 2 combinations, right? The top 2 are the same and the bottom 2 are the same. In independent assortment, what happens is that homologous pairs, they can align in different arrangements. So for example, let's look at another possible alignment. So now you can see that the chromosome pair, the homologous pair boxed in, now the blue chromosome, replicated chromosome from your dad is on top, and the red replicated chromosome from your mom is on the bottom. So after metaphase comes anaphase, where these chromosomes are separated, and basically at the end of meiosis, each haploid cell is going to get a random assortment of your mom and dad's chromosomes. So you can see now that we've created an additional combination of chromosomes where the top, now you have chromosome 1 from your mom and chromosome 2 from your dad, and on the bottom you have chromosome 1 from your dad and chromosome 2 from your mom. So basically you have 23 pairs of chromosomes, right? So you can get many arrangements of chromosomes. Basically, you're just shuffling which chromosomes are from your mom and which ones are from your dad. So you can get cells that have a mix of those 23 different chromosomes. And this creates diversity. And again, diversity or variation is a good thing. Okay, so let's end with how an error in meiosis can lead to different disorders. One of the most famous genetic disorders that can happen because of errors in meiosis is trisomy 21, which is also known as Down syndrome. But this is basically a human that has 3 chromosome 21s instead of 2 of them, right? We normally have 2 of each chromosome. People with Down syndrome have 3 of chromosome 21, only chromosome 21. And this is due to an error in anaphase 1. So let's see what is going on here. This is called non-disjunction. So you can notice there in metaphase 1, the homologous chromosome pairs have aligned at the center, and then in anaphase, that's going to separate the pairs, right? Well, you can see that in this case, one of the pairs didn't separate. So now you have both homologous chromosomes for what that would be chromosome 21 are going to end up in the top cell. So this is called non-disjunction. Disjunction basically means separation, right? Think of whatever that cartoon was, School Rock or something like that, but where they would have conjunction, junction, what's your function? So a conjunction is something that brings 2 things together. Disjunction means separation, right? Separating 2 things. Well, this is non-disjunction. This is where 2 things did not separate. So where chromosome 21, or you can have this can happen in different chromosomes and lead to different genetic disorders. But in this case, chromosome 21 did not go through disjunction. They did not separate. So chromosome 21 doesn't separate during anaphase 1, and that leads to trisomy 21 or Down syndrome. And here you have a cute little Down syndrome boy, very characteristic features of this genetic disorder. And so now you actually know or understand better how Down syndrome happens. It's just an extra chromosome 21. Okay, so we'll go over some of this stuff on Tuesday's lecture. I know it's a lot, and we'll clarify. We'll actually have a quiz on this stuff, so make sure you try and understand what's going on with the lecture. And yeah, we'll pick back up. Thank you guys for watching, and have an awesome weekend. Peace.